Breathing, also known as ventilation, is how the air moves into and out of the lungs. It consists of repetitive cycles of inspiration, when air flows into the lungs, expiration, when air leaves the lungs, and a brief pause, called the rest period, between these two. Now, the direction of airflow throughout the breathing cycle depends on the difference between the atmospheric pressure, which is the pressure of the air in the environment, and the alveolar pressure, or the pressure inside the alveoli, which are the tiny sacs of air where gas exchange happens in the lungs. An additional parameter is the intrapleural pressure, also called the intrathoracic pressure, which is the pressure of the fluid inside the pleural cavity that surrounds the lungs. Intrapleural pressure is usually negative compared to the alveolar or atmospheric pressure, and this is important because the alveolar pressure minus the intrapleural pressure gives the transmural pressure. As long as the transmural pressure stays positive, the airways remain open throughout all of the phases of the breathing cycle. Now, normal, quiet breathing involves inspiration and expiration of a tidal volume, or VT for short, of about 500 milliliters, which includes the volume of air that fills the alveoli plus the volume of air that fills the airways. Now, according to what is known as Boyle's law, at a constant temperature, pressure and volume are inversely related to each other. So, when the alveolar pressure decreases, more air will enter the lungs, increasing the air volume. With that in mind, let's establish the starting point for these variables by looking at the lungs during the rest phase of the breathing cycle. During rest, the diaphragm is at its balanced position. The alveolar pressure equals the atmospheric pressure to a value of 0 centimeters of water, so there is no pressure gradient, and no air is moving into or out of the lungs. The intrapleural pressure is negative, approximately negative 5 centimeters of water, because the lungs and the chest wall act as opposing forces, meaning the lungs have a tendency to collapse during rest, while the chest wall has a tendency to expand. Because alveolar pressure, which is 0 centimeters of water, minus the intrapleural pressure, which is negative 5 centimeters of water, equals a transmural pressure of plus 5 centimeters of water, that means that the airways are open during rest. Now, inspiration and a new breathing cycle start when there's a variation in the arterial pressure of oxygen, or PaO2, which is normally around 100 millimeters of mercury, the arterial pressure of carbon dioxide, or PaCO2, normally around 40 millimeters of mercury, and the arterial pH, which is normally 7.4. Changes related to these markers activate a series of receptors called chemoreceptors, which are specialized sensory cells that convert the concentration of a chemical substance in the blood, such as carbon dioxide or oxygen, to a biological signal for the respiratory center located in the brainstem. The respiratory center consists of three major respiratory groups of neurons. The dorsal respiratory group and the ventral respiratory group are found in the medulla oblongata, while the pontine respiratory group is found in the pons and consists of two areas, known as the pneumotaxic center and the apneustic center. Of these, the dorsal respiratory group, or DRG, is the one that initiates respiration, and it also determines the basic rhythm of breathing by adjusting the frequency of inspiration so as to keep the arterial pressure of oxygen, arterial pressure of carbon dioxide, and the arterial pH in the normal range. When the dorsal respiratory group receives information regarding the increase of arterial pressure of carbon dioxide, the DRG sends a command through the phrenic nerve to the diaphragm, which contracts to increase the vertical length of the thoracic cavity, and through the intercostal nerves to the external intercostal muscles, which contract and make the ribs move up and out, increasing the lateral size of the thoracic cavity. Based on Boyle's law, as lung volume increases, the alveolar pressure decreases, specifically to -1 centimeters of water. This is lower than the atmospheric pressure, so now there is a pressure gradient that makes the air rush into the lungs like a vacuum. Eventually, all this air brings the alveolar pressure back up, and once it equals the atmospheric pressure, airflow into the lung stops. At the same time, as lung volume increases, this compresses the intrapleural cavity, but the intrapleural pressure always remains lower than the alveolar pressure, reaching the value of -8 centimeters of water at the end of a normal inspiration. That means that the transmural pressure remains positive at a value of 8 centimeters of water, and the airways stay open. Thus, the volume present in the lungs at the end of normal inspiration is the sum of the one tidal volume and the volume of air left in the lungs at the end of the previous expiration, called the functional residual capacity, or FRC. Now, when oxygen requirements are higher, like during exercise, the apneustic center from the pons is activated. Stimulation of the neurons in the apneustic center excites the dorsal respiratory group in the medulla, prolonging the period of action potentials in the phrenic nerve and thereby prolonging the contraction of the diaphragm, leading to prolonged inspiration. Additionally, the external intercostal muscles found between the ribs and accessory muscles, such as the sternocleidomastoid and scalene muscles in the neck, also contract during vigorous inspiration, making the ribs move up and out. Finally, let's take the scenario to the extreme. Say you're exercising really hard and your apneustic center was so hyperstimulated that it was still trying to make you inhale when your lungs were already fully inflated. To prevent that, the pneumotaxic center activates, and it limits the burst of action potentials in the phrenic nerve, making the diaphragm contract less. This stops inspiration, allowing for expiration to happen so we can keep ventilating the lungs. Speaking of which, under normal circumstances, once the air movement stops and the inspiration ends, expiration begins. Normally, expiration is a passive process, meaning that there is no need for any group of neurons to stimulate muscle contraction. The diaphragm relaxes, and the air leaves the lungs due to their elasticity, which allows them to rebound after the inspiration like a filled balloon that lets the air out if you don't seal it after inflating. So essentially, the lungs squeeze back to their initial size and put pressure on the volume of air inside the alveoli. This makes the alveolar pressure increase to +1 centimeters of water, which is higher than the atmospheric pressure, creating yet another pressure gradient. But this time, the pressure gradient pushes the air out of the lungs, but about 500 milliliters of air, representing the FRC, remain inside the lungs to prevent the alveoli from shutting closed. Now the intrapleural pressure becomes less negative during expiration compared to inspiration, going back to a value of -5 centimeters of water. Basically, at the end of this phase, all volumes and pressures return to their values at rest, and a new breathing cycle begins. A particular situation is forced expiration, which is when a person forcibly breathes out, like a vocalist that is trying to hold a musical note. This is when the ventral respiratory group, or VRG, is activated. The VRG sends inhibitory impulses to the apneustic center, which has the role of increasing the duration of inspiration. So, when VRG inhibits the apneustic center, inspiration will be shorter, leaving more time for a longer expiration. Finally, all phases of the breathing cycle can be voluntarily controlled through commands from the cerebral cortex. For instance, a person can choose to hyperventilate, which means that the breathing frequency and volume will be increased, or hold their breath and thus hypoventilate. Okay, quick recap. Breathing involves inspiration, expiration, and a resting period between the two. Inspiration is the active phase when air enters the body, while expiration is the passive phase when air leaves the lungs. In order for this cycle to happen, there is a respiratory center which controls breathing. This center receives information from a group of receptors, known as chemoreceptors, which detect any changes regarding the pressures of the gases involved in breathing.
The primary role of the lungs is to ensure gas exchange between the external environment and the blood within the circulatory system. This happens thanks to a series of branching tubes called airways, which conduct the air down into small, thin-walled sacs called alveoli, which are wrapped in an intricate network of tiny blood vessels called pulmonary capillaries. And the alveolo-capillary membrane, where the layer of alveolar cells lining the alveoli meets the endothelial cells that make up the pulmonary capillary, is where gas exchange occurs. Now, before we delve into diffusion, perfusion, and their limits, remember that gas exchange across the alveolo-capillary membrane happens according to Fick's law. Fick's law states that the net rate of diffusion, V, of any particular gas across the alveolar capillary membrane is proportional to the pressure gradient across the wall, which is the difference between the partial pressure of the gas in the alveolar sacs, or P capital A, and the partial pressure of the gas in the blood, or P lowercase a, and also proportional to the surface area of the wall, or A, but inversely proportional to the wall's thickness, T. And this is all times the diffusion constant, D, which varies from gas to gas. So, diffusion-limited gas exchange means that a gas like oxygen or carbon dioxide can diffuse across the alveolar capillary membrane as long as the partial pressure gradient is maintained. On the other hand, perfusion-limited gas exchange means that if the pressure gradient is not maintained and the concentration of gases on the two sides of the alveolar capillary membrane become the same, further gas exchange is only possible by increasing blood flow, or perfusion, in the pulmonary capillary. To understand these concepts, let's look at a section of an alveolar sac, with a pulmonary capillary running along its surface. The capillary carries mixed venous blood, which is low in oxygen and high in carbon dioxide. As blood passes along the alveolar capillary wall along the length of the capillary, it exchanges some gas molecules with the interior of the alveolar sac. Let's look at perfusion-limited gas exchange first. In this case, the total amount of gas that diffuses into the blood depends on the amount of blood flowing through the pulmonary capillaries around the alveoli. The best example to illustrate this is by using nitrous oxide, or N2O gas. So, imagine we're inflating the alveolar sac with nitrous oxide, and that its partial alveolar pressure, noted as P capital A N2O, is constant. At the beginning of the alveolar capillary contact, the partial pressure of nitrous oxide in the pulmonary capillary blood, or P lowercase a N2O, is 0, because this gas doesn't initially exist inside the blood. According to Fick's law, this pressure gradient makes nitrous oxide molecules from the alveolar sac rapidly diffuse into the pulmonary capillary. Within the blood, nitrous oxide molecules don't bind to hemoglobin or other blood components, so all the gas molecules are free in the blood, which makes the partial pressure of nitrous oxide in the pulmonary capillary blood rise quickly. In fact, this happens so fast that by the first 1/5 of the capillary length, the equilibrium between the partial pressures on both sides of the alveolar capillary membrane is already attained. At this point, the partial pressure gradient falls to 0, so nitrous oxide diffusion stops. In this situation, the only way to increase the net diffusion is to increase blood flow through the pulmonary capillary. Increased blood flow rushes nitrous oxide-rich blood through the capillary and brings in new blood with no or less nitrous oxide in it, restoring the partial pressure gradient. Another example of perfusion-limited gas exchange can be illustrated by oxygen, but only under normal circumstances. So this does not apply during strenuous exercise or in the case of lung disorders like fibrosis or emphysema. Just your regular run-of-the-mill oxygen diffusion. Alright, so at the beginning of the alveolar capillary contact, the partial pressure of oxygen in the alveoli, or PAO2, is higher than that which comes within mixed venous blood in the pulmonary capillaries, which is noted as P_a_O2. This creates a partial pressure gradient, which makes oxygen diffuse into the blood. As the process continues, the partial pressure of oxygen in the pulmonary capillary blood rises, but not as quick as with nitrous oxide. And this is because, unlike nitrous oxide, newly diffused oxygen molecules bind to hemoglobin in red blood cells. So first, all hemoglobin molecules have to become saturated with oxygen before free oxygen molecules appear in the blood and the partial pressure of oxygen increases. This pushes the point of equilibrium between PAO2 and Pâ at around 1/3 of the capillary length, a bit further compared to the 1/5 in nitrous oxide. From here onwards, unless the rate of blood flow increases to bring in more oxygen-hungry blood, no more net diffusion of O2 can occur. Now, on the other hand, there's diffusion-limited gas exchange, which is best illustrated by using the example of carbon monoxide, or CO, diffusion. Okay, now, at the beginning of the alveolar capillary contact, we have the highest alveolar CO partial pressure, or PACO, while its partial pressure in the blood, or Pâco, is 0, because this gas isn't originally found in the blood. At this point, the partial pressure gradient is at its highest, and this causes carbon monoxide to quickly diffuse into the blood. When carbon monoxide gets into the blood, it avidly binds to hemoglobin within the red blood cells. So, there's very little free unbound CO. This causes partial pressure of carbon monoxide in the blood to rise just a little bit. In fact, it rises so little that even at the end of the capillary length, the equilibrium between alveolar carbon monoxide partial pressure and partial pressure of the carbon monoxide in the blood is not achieved. So the pressure gradient is sustained along the entire length of the pulmonary capillary, making this a case of diffusion-limited gas exchange. Diffusion-limited gas exchange also applies to oxygen under special circumstances, like during strenuous physical exercise. In this situation, heart rate increases, the speed of pulmonary blood flow increases, and this shortens the transit time for the blood near the alveolar capillary wall. So there's no time for partial pressure of oxygen in the blood to reach the partial pressure of oxygen in the alveolar sac. This keeps the pressure gradient up and running, so to speak, making it a type of diffusion-limited gas exchange. Oxygen exchange is also diffusion-limited in the setting of lung conditions like lung fibrosis or emphysema. In lung fibrosis, the alveolar capillary wall thickens, and according to Fick's law, a thicker alveolar capillary membrane reduces the rate of diffusion, so it takes much longer for the partial pressure of oxygen in the blood to reach the partial pressure of oxygen in the alveolar sac. This means that blood flows through the entire length of the capillary with a pressure gradient in place that's high enough to maintain diffusion. Finally, there's emphysema. With emphysema, the alveoli are gradually destroyed, which reduces the total surface area that allows gas exchange. And, according to Fick's law, if there's less surface area for gas exchange to occur, the rate of diffusion decreases. This maintains a high partial pressure gradient, so it's another example of diffusion-limited gas exchange. Alright, as a quick recap, diffusion-limited and perfusion-limited gas exchange are two instances that alter gas exchange across the alveolo-capillary membrane. With perfusion-limited gas exchange, the partial pressures across the alveolar capillary wall equilibrate before blood leaves the pulmonary capillaries, which results in a null pressure gradient. Increasing diffusion in this case requires increasing the rate of blood flow in pulmonary capillaries. On the other hand, with diffusion-limited gas exchange, the partial pressures across the alveolar capillary barrier do not equilibrate because the gas molecules bind to some blood components like hemoglobin. So there's very little free gas in the blood, which exerts a minor partial pressure, too small to balance the one in the alveolar sacs. This sustains the pressure gradient along the entire length of the pulmonary capillary, making it diffusion-limited gas exchange.
Gas exchange is the physical process by which gases move passively, meaning that no energy is required to power the transport by diffusion across a surface. External respiration is another term for gas exchange. It describes both the bulk flow of air into and out of the lungs and the transfer of oxygen and carbon dioxide into the bloodstream through diffusion. Internal respiration, on the other hand, describes the capillary gas exchange in body tissues. While the flow of air from the external environment happens due to pressure changes in the lungs, the mechanisms of alveolar gas exchange are more complex. The primary three components of gas exchange are the surface area of the alveolo-capillary membrane, the partial pressure gradients of the gases, and the matching of ventilation and perfusion. So, if we were to draw a path for the oxygen molecules entering the body, it would start from the nose or mouth and end up in the lungs, where it reaches the alveoli, which are wrapped in an intricate network of tiny blood vessels called pulmonary capillaries. So from the alveoli, the gas molecules will go into the blood in the capillaries. Carbon dioxide follows the same path, but in the opposite direction, moving from the blood in the capillaries to the air in the alveoli and then getting exhaled. Now, the important role in this process belongs to the alveolo-capillary membrane. That's where the layer of alveolar cells lining the alveoli meets the endothelial cells that make up the pulmonary capillary. And this is where gas exchange happens. With that in mind, let's just say that when it comes to the surface area of the alveolo-capillary membrane, bigger is better, because a respiratory membrane with a large surface area has more gas to diffuse across it in a given period of time, leading to a more efficient gas exchange. With emphysema, for example, which is a condition where the alveoli are gradually destroyed, the total surface area that allows gas exchange is reduced. If there's less surface area for gas exchange to occur, the rate of diffusion decreases. Another aspect related to the alveolo-capillary membrane which influences gas exchange is its thickness. So in healthy lungs, respiratory membrane is 0.5 to 1 micrometer thick. In lung fibrosis, on the other hand, the alveolar capillary wall thickens, and a thicker alveolo-capillary membrane reduces the rate of diffusion. Now, the gas exchange across the alveolo-capillary membrane happens according to what is known as Fick's law. Fick's law states that the net rate of diffusion, V, of any particular gas across the alveolar capillary membrane is proportional to the driving force, which is the difference between the partial pressure of the gas in the alveolar sacs, or P capital A, and the partial pressure of the gas in the blood, or P lowercase a, and also proportional to the surface area of the membrane, or A, but inversely proportional to the wall's thickness, T. And this is all times the diffusion coefficient, D, which varies from gas to gas. Therefore, V equals P capital A minus P little a times D divided by T. Specifically, the driving force for diffusion is the partial pressure difference of the gas across the membrane and not the concentration difference. So the diffusion of oxygen and carbon dioxide are driven across the respiratory membrane by their partial pressure gradients. Therefore, if the oxygen partial pressure in alveolar air is 100 millimeters of mercury, and the one of mixed venous blood entering the pulmonary capillary is 40 millimeters mercury, then we have a driving force for oxygen across the alveolar capillary barrier of 60 millimeters of mercury. Basically, a steep oxygen partial pressure gradient occurs through the alveolo-capillary membrane because the partial pressure of oxygen in the alveolar air is greater than the partial pressure of oxygen in the pulmonary arteries, causing oxygen to rapidly cross the respiratory membrane from the alveoli into the blood. The partial pressure of carbon dioxide is also different between the alveolar air and the blood of the capillary. However, the partial pressure difference is less than that of oxygen, about 5 millimeters of mercury. The partial pressure of carbon dioxide in the blood of the capillary is about 45 millimeters of mercury, whereas its partial pressure in the alveoli is about 40 millimeters of mercury. Now, the partial pressures of inhaled air and alveolar air determine why oxygen goes into the alveoli and why carbon dioxide leaves the alveoli. This brings us to what is called Dalton's law, which states that the sum of partial pressures of all gases in a mixture equals the total pressure of that mixture. Thus, for dry gas, the partial pressure is the total pressure multiplied by the fractional concentration of dry gas, while the relationship for humidified gas is determined by correcting the barometric pressure for the water vapor pressure. Therefore, for dry gas, Px equals Pb times F. For humidified gas, Px equals Pb minus pH2O times F, where Px is a partial pressure of a gas, Pb is a barometric pressure, pH2O is the water vapor pressure at 37 degrees Celsius, 98.6 degrees Fahrenheit, having a value of 47 millimeters of mercury. F is a fractional concentration of a gas. To exemplify this, in dry inspired air, the partial pressure of oxygen is approximately 160 millimeters of mercury, which is computed by multiplying the barometric pressure of oxygen, which is 760 millimeters of mercury, by the fractional concentration of oxygen, 21%. For practical purposes, there is no carbon dioxide in dry inspired air, and partial pressure of carbon dioxide is 0. In humidified tracheal air, it is assumed that the air becomes fully saturated with water vapor. At 37 degrees C, partial pressure of H2O is 47 millimeters of mercury. Thus, in comparison to dry inspired air, humidified tracheal air has a lower partial pressure of oxygen because the oxygen is diluted by water vapor. Again, recall that partial pressures in humidified air are calculated by correcting the barometric pressure for water vapor pressure, then multiplying by the fractional concentration of the gas. Thus, the partial pressure of oxygen of humidified tracheal air is 150 millimeters of mercury. Because there is no carbon dioxide in inspired air, the partial pressure of carbon dioxide of humidified tracheal air is also 0. The humidified air enters the alveoli where gas exchange occurs. In alveolar air, the values for partial pressure of oxygen and partial pressure of carbon dioxide are changed substantially when compared with inspired air. Partial alveolar pressure of oxygen is 100 millimeters of mercury, which is less than the partial pressure of oxygen in inspired air, which is 160 millimeters of mercury. And partial alveolar pressure of carbon dioxide is 40 millimeters of mercury, which is greater than the partial pressure of carbon dioxide in inspired air, which is close to 0 millimeters of mercury. So according to Dalton's law, these partial pressure values influence the moving of these gases, meaning they will move from an area of high concentration to an area of low concentration. Now, within the lungs, oxygen and carbon dioxide diffuse between the air in the alveoli and the blood, that is, between a gas and a liquid. This movement is governed by Henry's law, which states that at a constant temperature, the amount of gas that dissolves in a liquid is directly proportional to the partial pressure of that gas in equilibrium with that liquid. Basically, according to this law, gases can be forced to dissolve into a liquid, let's say blood, if there is enough pressure applied and a controlled volume. It also says that once the pressure is released, gases can come out of solution. The concentration of a gas in solution is expressed as volume %, or milliliters of gas per 100 milliliters of blood. Thus, for blood, Cx equals Px times solubility, where Cx is the concentration of dissolved gas, that is, milliliters of gas per 100 milliliters of blood, Px is the partial pressure of gas, millimeters of mercury, and solubility refers to the solubility of gas in blood, and that's milliliters of gas per 100 milliliters of blood per millimeters of mercury. So, to test this relationship, let's consider the partial pressure of oxygen of arterial blood is 100 millimeters of mercury. And given that the solubility of oxygen is 0.003 milliliters of oxygen per 100 milliliters of blood per millimeter of mercury, we should be able to know what is the concentration of dissolved oxygen in blood, right? Well, yes, because oxygen equals partial pressure of oxygen times solubility. So the concentration of dissolved oxygen is 0.3 milliliters per 100 milliliters of blood. Actually, this law interconnects with Boyle's law during breathing cycle and gas exchange. Boyle's law states that for a fixed amount of a gas kept at a fixed temperature, pressure and volume are inversely proportional. Basically, if volume increases, then pressure decreases, and the other way around. This law has applicability in the respiratory system during inspiration and expiration. In inspiration, lung volume increases, which decreases alveolar pressure, creating a negative pressure gradient that allows oxygen to flow into the lungs. In expiration, the alveolar pressure increases and lung volume decreases, making the air exit the lungs. Correlating this information with Henry's law, we can conclude that in inspiration, carbon dioxide will return to its gaseous state because the alveolar pressure is decreased and it cannot push it into the blood. Less carbon dioxide in the blood makes a smaller concentration gradient between blood and alveoli space, so it becomes harder for gas exchange to happen. On the other hand, during expiration, the volume is decreased and the alveolar pressure is increased. That will force oxygen into blood because pressure pushes it there. More oxygen into blood creates a larger concentration gradient, and oxygen exchange is more efficient. At rest, the time that it takes for the diffusion of carbon dioxide from the pulmonary capillary blood into the alveoli is about the same as that of oxygen, because the solubility of carbon dioxide is much greater than that of oxygen, by a factor of about 20 in both blood and alveolar fluids. All right, so in order to understand Henry's law better, let's consider emphysema again, where the alveoli or the air sacs of the lungs are damaged. This damage usually means that air is trapped in there. In terms of volume and pressure, trapped air means a constant high volume and a constantly low pressure. As a result, in emphysema, carbon dioxide will easily diffuse out of blood, but oxygen will not be forced into blood, and that will lead to hypoxemia or low partial pressure of oxygen. An everyday example of Henry's law is carbonated soft drinks. Before the bottle or can is opened, the gas above the drink is almost pure carbon dioxide at a pressure slightly higher than the atmospheric pressure. The drink itself contains dissolved carbon dioxide. When the bottle or can is opened, some of this gas escapes, giving the characteristic hiss. Because the pressure above the liquid is now lower, some of the dissolved carbon dioxide comes out of solution as bubbles. If a glass of the drink is left in the open, the concentration of carbon dioxide in solution will come into equilibrium with carbon dioxide in the air, and the drink will go flat. Now, the last puzzle piece for an optimal external respiration is to have a close match between ventilation, aka the amount of gas reaching the alveoli, and perfusion, which is the blood flow in pulmonary capillaries, creating a balance. So, when alveoli have high partial pressure of oxygen and low partial pressure of carbon dioxide, the bronchioles serving the alveoli constrict because there isn't much carbon dioxide that needs to be removed. And the pulmonary capillaries dilate, allowing the blood to pick up oxygen. On the other hand, when the alveoli have low partial pressure of oxygen and high partial pressure of carbon dioxide, the bronchioles serving the alveoli dilate because the carbon dioxide needs to be removed, and the pulmonary capillaries constrict because there's relatively little oxygen to be picked up, and that allows blood to go to other alveoli that may have more oxygen. Therefore, partial pressure of carbon dioxide controls ventilation by changing bronchiolar diameter, and partial pressure of oxygen controls perfusion by changing arteriolar diameter. Now, since we discussed all the factors which influence external respiration, let's check if we got it right as to what happens during this process. So, the mixed venous blood returns from the tissues via the veins to the right heart. It is then pumped from the right ventricle into the pulmonary artery, which delivers it to the pulmonary capillaries. The composition of this mixed venous blood at this point reflects metabolic activity of the tissues. The partial pressure of carbon dioxide level in the blood is greater than the partial pressure of oxygen level at a value of 45 millimeters of mercury, because the tissues have produced carbon dioxide and added it to venous blood. The partial pressure of oxygen in the blood is at 40 millimeters of mercury, because the tissues have taken up and consumed oxygen. On the other hand, the oxygenated blood leaving the pulmonary capillary is delivered to the left heart and becomes systemic arterial blood. The oxygenation is affected by the exchange of oxygen and carbon dioxide between the alveolar air and pulmonary capillary blood. Because diffusion of gases across the alveolo-capillary membrane is fast, blood leaving pulmonary capillaries normally has the same partial pressure of oxygen and partial pressure of carbon dioxide as alveolar air, meaning there is complete equilibration. Hence, partial arterial pressure of oxygen is 100 millimeters of mercury, and partial arterial pressure of carbon dioxide is 40 millimeters of mercury, just as partial alveolar pressure of oxygen is 100 millimeters of mercury, and partial alveolar pressure of carbon dioxide is 40 millimeters of mercury. This arterialized blood will now be returned to the left heart, pumped out of the left ventricle into the aorta, and begin the cycle again. Now, there is a small discrepancy between alveolar air and systemic arterial blood. Systemic arterial blood has a slightly lower partial pressure of oxygen than alveolar air. This discrepancy is a result of a physiologic shunt, which allows a small amount of pulmonary blood flow to bypass the alveoli and skip oxygenation. The physiologic shunt has two sources, bronchial blood flow and a small portion of coronary venous blood that drains directly into the left ventricle rather than going to the lungs to be oxygenated. When the physiologic shunt in pulmonary blood flow is increased, it is called a ventilation perfusion defect. If the size of the shunt increases, equilibration between alveolar gas and pulmonary capillary blood cannot adequately occur, and pulmonary capillary blood is not fully oxygenated. What is known as the AA difference expresses the difference in partial pressure of oxygen between alveolar gas, capital A, and systemic arterial blood, lowercase a. If the shunt is small, which defines it as physiologic, then the AA difference is small or negligible. If the shunt is larger than normal, then the AA difference increases to the extent that oxygen equilibration fails to occur. Now let's switch gears to internal respiration, where the partial pressures and diffusion gradients are reversed from the situation described in external respiration. However, the factors that influence tissue gas exchange are similar to the factors of alveolar gas exchange and include partial pressure gradients between the blood and tissues, the blood perfusion of those tissues, and the surface area of those tissues. So during internal respiration, the partial pressure of oxygen in tissues is low, at about 40 millimeters of mercury, because oxygen is continuously used for cellular respiration. In contrast, the partial pressure of oxygen in the blood is about 100 millimeters of mercury. This creates a pressure gradient that causes oxygen to dissociate from hemoglobin, diffuse out of the blood, cross the interstitial space, and enter the tissue. Hemoglobin that now has little oxygen bound to it loses much of its brightness, so that blood returning to the heart is more burgundy in color, in contrast to external respiration, where dark red deoxygenated blood from the pulmonary circuit becomes bright red oxygenated blood to intersystemic circulation. On the other hand, considering that cellular respiration continuously produces carbon dioxide, the partial pressure of carbon dioxide is lower in the blood than it is in the tissue, causing carbon dioxide to diffuse out of the tissue, cross the interstitial fluid, and enter the blood. It is then carried back into the lungs, either bound to hemoglobin, dissolved in plasma, or in a converted form. By the time blood returns to the heart, the partial pressure of oxygen has returned to about 40 millimeters of mercury, and the partial pressure of carbon dioxide has returned to about 45 millimeters of mercury. The blood is then pumped back to the lungs to be oxygenated once again during external respiration. Okay, quick recap. Gas exchange between blood and alveoli, as well as between blood and tissue cells, takes place by simple diffusion. At the respiratory membrane, where the alveolar and capillary walls meet, gases move across the membrane, with oxygen entering the bloodstream and carbon dioxide exiting. Besides the surface area of the alveolo-capillary membrane, the partial pressure gradients of the gases and the matching of perfusion and ventilation are vital for the exchange to happen.